Dr. Sage here, back to continue our discussion of microbial genetics. In today's video, we're going to talk about mutations, which are changes in the genetic code. By the end of this video, you should be able to define the term mutation and discuss one positive and one negative example of it in microorganisms. You should also be able to differentiate among frameshift, nonsense, silent, and missense mutations. First, what is a mutation? It's any change to the nucleotide sequence in the genome. It is the driving force of evolution. In microorganisms, mutations become evident in altered gene expression, such as altered pigment production or development of resistance to a drug. A couple more definitions. Wild type is a microorganism that exhibits a natural, non-mutated characteristic and is the trait present in the highest numbers in a population. A mutant strain is an organism that bears a mutation. What are the causes of mutations? We can have spontaneous mutations, which is a random change in the DNA arising from errors in replication that occur randomly. In other words, the DNA replication machinery, like DNA polymerase, does a great job of copying the DNA. However, it's not perfect. Once in a while it makes a mistake and puts the wrong base. Or you can have induced mutations. These result from exposures to known mutagens. For example, in humans, cigarette smoke. Cigarette smoke contains mutagens. When you breathe it into your lungs, it can mutate the DNA of your lung cells, and that leads to lung cancer. So, some selected mutagenic agents and their effects. Well, we can have chemical agents, or we can have radiation agents. What are the categories of mutations? Well, we can have a lethal mutation. That's a mutation that leads to cell dysfunction or death. We can have a neutral mutation that produces neither an adverse or a helpful change. There's also a back mutation. That occurs when a gene that has undergone a mutation reverses to its original base composition. So with mutations, there's different types. One category that we're going to talk about today are called point mutations. Point mutations are small mutations that affect only a single base on a gene. They involve addition, deletion, or substitution of single bases. So point mutation is a small mutation. It changes one single point, like one base pair, for example. With the point mutations, there are two types. You can have substitutions. That's where, let's say, A, for example, is turned into a C. That would be a substitution. Or you can have insertions or deletions. So let's say you had like C, A, T, T in your base code, and then you had an extra base, like let's say a G, added. That'd be insertion. Or if you lost a base, like you lost the T, that'd be a deletion. Okay, so what effects can these point mutations have? Well, one effect is a silent mutation. So on the left here, we have the normal DNA sequence. We can see here for some gene. That is transcribed into mRNA, and that is translated into a polypeptide or protein. Well, let's say we have a point mutation that is a substitution. This A is turned into this T. So it is a mutation. There is a change to the DNA, but there's actually no change to the protein. So recall the genetic code is redundant. So this codon, GUA, codes for the amino acid valine. Well, this A was turned into a T, which means it turns the codon GUA into the codon GUU. But both GUA and GUU, they both code for valine. So even though there is a mutation, there's a change to the DNA, there is no change to the protein. The protein is exactly the same. That's called a silent mutation. So a silent mutation is a change to the DNA, but no change to the protein. Next, we can have a missense mutation. So again, let's say we have a point mutation, so a single base pair change. Again, let's say it's a substitution. This C is turned into this A. This C was part of the codon CCC, which codes for the amino acid proline. This A is now part of the codon ACC, which codes for the amino acid threonine. In other words, one amino acid has been turned into one different amino acid. That is called a missense mutation. Another example is called a nonsense mutation. So again, let's say we have a point mutation. Again, let's say we have a substitution. This base C is turned into this base G. That changes this codon UAC, which codes for threonine, into this codon UAG, which is a stop codon. So what a nonsense mutation is it creates an early stop codon. What that means is you're not making the rest of the protein. Okay, if this protein were 1,000 amino acids long and you create a stop codon at the 4th amino acid, that means you don't have the whole rest of the protein, which means the protein is definitely not going to be functional. So if you get a nonsense mutation or an early stop codon near the beginning of a gene, it's one of the things that's most likely to cause that protein to stop functioning. The other type of mutation you can get comes from insertions and deletions instead of substitutions. Okay, this type of mutation is called a frameshift mutation. It occurs when one or more bases are inserted into or deleted from a newly synthesized DNA strand. This alters the reading frame of the mRNA, and it nearly always results in a non-functional protein. Every amino acid after the mutation is different from what is coded for in the original DNA. Insertions of bases in multiples of 3, however, do not disturb the reading frame. Alright, so let me explain that a little more detail. Recall that the way that the DNA code is read is kind of like letters forming words in a sentence. But whenever you read your words in a sentence, remember how you read it is there's spaces between the words. There's no spaces in the DNA. So how the DNA is read is in 3-letter long words called codon. So for example, let's say I have some letters here, and I don't have any spaces between them. Okay, but I tell you, read 3 letters at a time. In that case, you'll read dog, ate, cat. Okay, and then you can form a sentence. Even if there's no spaces, if I tell you read 3 letters together, and then the next 3, and then the next 3, then you'll know how to read the sentence, and that's called reading within the reading frame. Let's say something happens. Let's say you have a deletion, so one of the bases is removed. Let's say remove this base here, A. Now if you remove that base A, remember there's not actually a space left anymore. So how does it read it? Well, it still reads the 3 letters dog, but then it reads the next 3 together, tech, and then at, and then whatever else comes after that. In other words, you've shifted the reading frame. Even though the letters C-A-T are actually still there, you can't read the word cat anymore because you shifted the reading frame. Okay, so let's back up. Let's again put what we had originally, dog, ate, cat. And let's say instead of a deletion, let's say we have an insertion. Let's say that a G base is added here. Now, again, how do you read it? 3 letters together. Dog, gat, eka, and then whatever else comes after that. Again, even though the letters A-T-E and C-A-T are there, you cannot read the words ate cat because you've shifted the reading frame. Which means every word after that is going to be the wrong word. Or, as actually applicable to us, every amino acid after that is going to be the wrong amino acid. So, if you insert or delete 1 or 2 base pairs, it causes a frameshift mutation, and every amino acid will be the wrong amino acid after that. Now, if you happen to insert or delete 3 base pairs, in that case it doesn't shift the reading frame, it just like removes a single amino acid or adds an amino acid. In that case, if I erase the letters A-T-E, you can still read both dog and cat. So here we have an example. So we have a point mutation, but this time it's going to be a deletion. So these bases TA, which were part of this codon GUA to make valine, are removed. So now, instead of having GTA, you have GCC, which changes that amino acid and every amino acid after that, because it's shifting the reading frame. Okay, so mutations occur. They occur naturally and by things we're exposed to, but we and bacteria have ways of repairing mutations. One method is photoactivation. That's light repair of damage caused by ultraviolet radiation. Another type of repair is excision repair. Mutations are excised by a series of enzymes that remove the incorrect bases and add the correct bases. Or we can also have mismatch repair. Mismatch repair is a repair system that can locate mismatched bases that were missed during proofreading. The base must be replaced soon after the mismatch is made or will not be recognized by the repair enzymes. So how do we know if a chemical is a mutagen, if it can mutate DNA? Well, one way is by a method called the Ames test. This is commonly used to rapidly detect chemicals with carcinogenic potential. This uses bacteria instead of experimental animals. It allows easy observation and monitoring of gene expression and mutation rate. Any chemical capable of mutating bacterial DNA could similarly mutate mammalian DNA and is potentially hazardous. So this is one way of us identifying if a chemical is a mutagen, if it can mutate our DNA, which means that chemical can be a carcinogen, can lead to cancer. So what happens is they have a strain of bacteria, Salmonella, that requires histidine to live. If there's no histidine present in the media they're growing on, then they can't live. So you set up this Salmonella strain as a negative control without a possible mutagen, and the same Salmonella strain with a possible mutagen. Okay, if this mutagen is mutating the DNA of the Salmonella, then it can mutate the Salmonella to no longer require them to have histidine in its media. In other words, they can produce their own histidine. So if you get more colonies forming than without the mutagen, what's happening is the mutagen is mutating the DNA of the Salmonella, allowing them to survive on this media where they naturally wouldn't be able to survive. This then is telling you that a chemical can mutate DNA and could be a carcinogen to humans. Mutations can have both positive and negative effects. Many mutations are not repaired. Effects of mutations depend on the nature of the mutation and the strategies available to the organism. Mutations are permanent and heritable. They're passed on to the offspring of organisms and new viruses and become a long-term part of the new gene pool. A small number of mutations contribute to the success of individuals and the population. Variant strains with alternate ways of expressing a trait can more readily adapt, survive, and reproduce. So, this has been your brief introduction to mutations, which are changes in the genetic code. Until next time, this has been Dr. Sage.